Hello, everyone. Welcome to today's webcast. Thank you so much for joining us. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix, and today I have the honor to introduce Dr. Rohan Palmer of Brown University. Dr. Palmer was one of our winners in last year's Abstract Challenge, and today he will present his abstract submission, Investigating Shared Additive Genetic Variation for Alcohol Dependence. Rohan, I'll hand it over to you.
Thank you, Cheryl, and thanks for everyone who came today to the talk. I'm going to start by giving a brief outline of today's presentation. First, we're going to talk about the consequences and potential mechanisms of alcohol harm, and then proceed to talking about the approach that we used in this study to describe genome-wide comparison of effects across ancestral populations, primarily individuals of European and African ancestry. I'm then going to summarize findings from this study and talk a bit about future directions that we're pursuing in this lab. And hopefully if we have time at the end, and depending on the number of questions, we might address a few of them. So alcohol misuse, so you might know, is actually a global concern, primarily because of the high rates of use throughout the world. Regular alcohol consumption is considered a major risk factor for increased mortality, and a good example of that really is the fact that we see roughly about 3.3 million deaths being attributable to alcohol consumption in the world, at least as of 2012. Within the United States, alcohol is actually currently the fourth leading cause of death. Now, there are multiple mechanisms of alcohol harm. Primarily, the three that I'd like to talk about and focus on today are really the toxic effects that can come from alcohol use, which, of course, those are the effects that would occur due to damage from chronic use. And so, for example, typically what you see is that excessive alcohol use can actually cause the pancreas to produce toxic substances that can interfere with proper functioning. Other things we want to be concerned about really is that the fact that if a woman drinks alcohol while she's pregnant, it may actually harm her developing baby. We can see that alcohol can actually pass from the mother's blood into the baby's blood, and it can damage and affect the growth of the baby's cells. Now, with regards to intoxication, we do know that alcohol can actually lead to impairment in both physical coordination, consciousness, and cognition. A good example of that being sort of deterioration in memory performance over time. And of course, there are clear of that when we think about drinking and driving, right? But what we're really going to focus on today is sort of the clinical definition of what we call alcohol dependence. And that's really talking about where individuals' loss of control over his or her drinking behavior. And to be more definitive about what I mean by the clinical definition of alcohol dependence, we're really talking about the DSM-IV criteria for alcohol dependence, which is really described as this maladaptive pattern of alcohol abuse, which leads to impairment in several sort of physiological and psychological domains. And these are shown to the right. And typically what we see in the clinical world, to meet the DSM-IV criteria for alcohol dependence, you have to really report having presented with 3 or more of these symptoms within the past 12 months, and those being sort of tolerance, withdrawal, using alcohol longer than intended, being unable to cut down or quit drinking alcohol, spending a lot of time using alcohol or recovering from the effects of alcohol, of course then foregoing several social or occupational activities, and of course drinking despite having physiological or psychological problems. Some of you might have also heard that the DSM criteria for alcohol dependence or alcohol use disorders has recently changed to go up to about 11 different criteria, and that's called the DSM-V. We won't be talking about studies looking at that today, but these are the different criteria that we're going to be looking at. So in regards to the genetics of alcohol dependence, what we've learned over the past several decades is that alcohol dependence tends to run in families, and generally what's seen is that if you have an alcohol-dependent relative, your risk tends to be quite elevated as a function of how close that relative is to you. Now, with respect to twin and adoption studies, what we have seen is that, by and large, genetic differences within families and within general population tend to account for about half of the risk for alcohol dependence. What we typically find is that genetic differences can either increase a person's risk or decrease a person's risk. But we want to make it clear that there really is no such thing as sort of an alcoholism gene. Alcohol dependence is influenced by many genetic factors that are responsible for normal biological functioning. And it's really the case, what we're trying to understand really is where variation in that functioning that is encoded by our genes makes us susceptible to the effects of alcohol. And of course, the thing to keep in mind is that as a disease, alcohol is influenced by both genetic factors and environmental factors, things such as our familial and sociocultural factors that also contribute to the disease and often interact with your genome to determine your risk for the disorder. Now, one thing to keep in mind is that once an individual starts to use alcohol, their risk for developing alcoholism or alcohol dependence is going to be greatly influenced by their genetics at that particular point in time. So in thinking about the reason why alcohol dependence is so etiologically complex or sort of genetically complex, I think it's important to talk about sort of the different types of genetically influenced diseases. And alcohol dependence typically falls within the category of complex disease. And one of the ways to interpret that really is to make a contrast between complex diseases and monogenic diseases like Huntington's disease. Now, in the case of Huntington's, typically what we see is a clear pattern of familial inheritance, right? But we also see that the genetic variant of interest that's being co-transmitted with disease has a very large effect on your likelihood of having that disorder. In contrast, complex diseases like alcohol dependence are influenced by many genetic loci, and these genetic loci each have a small effect or indirect effect on the outcome. And so the effect sizes typically are quite small. And the challenge there in that is that it's very difficult for us to begin to localize or identify those individual variants in our genome that are affecting your risk for having the disease. So to give you a good sense of that, what I want to show you really is sort of a summary of a genome-wide association study that was conducted by Andrew Heath and colleagues out of Washington University back in 2011. And really what the figure on the left is showing you is what we call a Manhattan plot. And it's summarizing the association analysis that was done across sort of the millions of variants that were tested, individual loci that were tested within that Australian sample that was used. And typically what we set as a threshold for genome-wide association studies are 5 × 10 to the -8. And so on the y-axis here for this graph, that would essentially be represented as expressed in terms of negative log 10 p-values. And basically what you see here is that no particular allele meets that particular threshold. But what they do understand is that as a twin study analysis, you can actually account for some of the variability, but identifying the individual loci tends to be quite difficult to resolve. Now, that being the case, we still know that individual loci contribute, or individual genes contribute to risk for alcohol dependence. And a good example of that really comes from the list of genes on the right-hand side of the slide, where we can identify variation within aldehyde dehydrogenase or the mu-opioid receptor systems that can actually be manipulated pharmacologically to reduce a person's risk of going on to use alcohol heavily. So what I want to summarize here also is a study that we recently published last year, which actually looked at the different criteria that contribute to alcohol dependence. And the reason why I want to present this is because in addition to being a clinical disease, one of the ways we can define alcohol dependence is by the different criteria that are being endorsed. And typically one of the things that we're concerned about is whether or not individuals are endorsing the same kinds of criteria. And so when you come up with these sort of clinical outcomes, sort of yes, no, you have a disorder, one of the things we have to worry about really is whether or not there's artificial heterogeneity that's being created in our analysis that's driving down our power to do so, to identify genetic loci. And so what we wanted to do was to actually demonstrate that there is evidence to suggest that genetic loci contribute to the individual criteria for the disease. But at the same time, if you look across the genetic influences, it's also possible to identify and determine that the majority of the effects that you're seeing on any one symptom of disease actually overlaps with another symptom. And what this suggests to us is that looking at alcohol dependence as a clinical outcome is useful, as well as looking at it as a sort of a continuous outcome, whether or not you want to sum across symptoms or do what we call exploratory and confirmatory factor models to come up with a score. And so generally what's seen when you do those kinds of analyses is that it's looking at composite measures like diagnosis or looking at continuous outcomes like factor score, in that we find that variation across our genome can account for roughly about 30% of the individual differences in the disease. Likewise, with respect to the individual items, we can see that genetic variation actually accounts for anywhere from 7 to roughly about 30% of the variance in any one symptom. And that's useful because it tells us that loci are important. But at the same time, we know that it's difficult for us to localize those individual loci, right? And there are many reasons for that. Some of them being, of course, that the effect sizes are so small that we need very large samples in order to be able to detect them. And of course, as we just talked about, this idea of definition of clinical phenotypes may actually lack disease sensitivity. And really what we're talking about there really is the fact that you can go on to use, but we're not necessarily measuring the changes that take place in an individual as they use over time. We're focusing on these clinical outcomes that are more distal to the disease of interest. Another point really is that there's a sort of failure to model all SNPs simultaneously, and that's been the challenge that's being overcome gradually with the implementation of mixed linear models more and more in association analyses. But the beautiful thing about that is that we're getting more and more less biased SNP effects by using those kinds of models, and since it's only recently been taking off, we haven't really been getting a good approximation of what the SNP effects are actually like. And of course, there's this bias within genome-wide association literature, not just for alcohol, but for many other disorders, in the sense that most of the studies tend to be focused on individuals of European descent, and we understand that, given the differences in allele frequencies across different ancestral populations, there are going to be certain gains in power or lack thereof to actually test certain individual loci across certain regions. And so it is to our advantage to look across different populations to see whether or not we can tap into some source of genetic variance. So the study that I'm talking about today is actually not the first to look at individuals of European and African ancestry when trying to understand the role of genetic factors in alcohol dependence. There was a nice study that came out in 2014 by Joel Galertner and colleagues out of Yale, which actually conducted a large genome-wide association study using about 16,000 individuals. Now, in that study, they actually used a large sample of imputed SNPs and used a significance threshold of 5 times 10 to the negative 8. Now, as sort of the basis for this study, their findings identified novel loci, but also confirmed, the ADH gene cluster on chromosome 4. And within that cluster, they were able to determine that there's evidence for biological convergence of loci that are influencing alcohol dependence. And this was the case for individuals of European and African ancestry. And of course, they did also note that there were some differences in SNPs, within those same regions, but it's evidence for convergence because a lot of those areas are sort of overlapping. And just to give you a sense of what the results look like from that study, as you can see here, the results for individuals of African ancestry, you see there's sort of this sort of clustering of some effects that reach that 5 times 10 to the negative 8 threshold here on chromosome 4. You can also see some nominally significant SNPs across other chromosomes as well. And with respect to individuals of European ancestry, for their case control analyses, we see sort of similar effects on chromosome 4, some nominal effects here on chromosome 3, as well as, and of course, significant effects on chromosome 12. And so for the current study, what I was really interested in understanding was the extent to which a lot of genetic effects we see genome-wide are actually overlapping between individuals of European ancestry, because we didn't really get that much of a complete picture from the Galertner et al. Study from 2014. And so to kind of summarize the individual goals, we really wanted to investigate the extent to which the additive genetic effects that we see contribute to each of the different aspects of alcohol dependence. And so we can think about additive genetic factors influencing tolerance, withdrawal, using longer than intended, failure to cut down or quit, great time using and recovering, activities forgone, and this use despite physiological or psychological problems. So of course, are there additive genetic effects across the genome on these individual criteria? We've done that before. Do they actually overlap, right? And is it actually the case that when you fit the model across individuals of European and African ancestry, that you find that that itself is correlated? So what we're really saying then is there evidence for a common set of genetic influences influencing the individual criteria for individuals of European ancestry? Do we see the same for individuals of African ancestry? And is there a genetic correlation operating between the two? And so what I'd like to do now is to transition to talking about the methods that we use. By and large, we're going to be trying to understand this black box that sort of contributes to data manipulation and imputation that goes into doing these kinds of studies. And it's always difficult to do this within the context of papers because there's that lack of space, right, to do so. And so in talking about study samples, we use a sample of roughly 20,500 individuals that were obtained from the database for genotypes and phenotypes. As you can see here, these studies came from the heroin on GWAS on heroin dependence, the Australian GWAS on alcohol dependence, the alcohol dependence GWAS in European and African Americans, as well as a study of addiction, genes, and environment. And primarily, all of these studies used a variety of different genotyping arrays. And so it was important for us to identify sort of the union of SNPs across these arrays to try to bring them up to a common place. So we're going to be spending a lot of time talking about how we achieved that, and that will facilitate anyone else trying to do the same thing with different samples or trying to replicate what we conducted for this study. So for all the analyses, we focused on alcohol dependence outcomes. What I'm going to be talking about today really are our analyses looking at the individual criteria. And for our models, we limited all our responses to individuals who actually been exposed. And so you actually have to report a history of actually using alcohol. And we didn't prohibit you from participating in the study if you actually didn't have a lifetime diagnosis of the problem. We also didn't exclude individuals if they had problems on other substances as well. So to give you an overview of how we manipulated data, we did it in 2 steps. The first step being largely data preparation in order to get the sample and the data in a place where we can actually impute it. And you can see here, the red boxes kind of summarize it. We start off, of course, with admixed populations of individuals from the dbGaP samples. Our goal really is to identify and separate the largest ancestral groups, which are those of individuals, which are those of European and African ancestry. We then go on to take those individual populations and impute them up to, phase 3, 1,000 genomes, and then conduct our analyses using sort of a pool set of SNPs across the different arrays. And so for step 1, we're really identifying the target variants across the different platforms. We then, of course, need to import our 1,000 genomes reference data, and you can get that from the Impute2 website. Of course, we then want to prep our 1,000 genomes data for Ancestry Determination within SVS, checking strand orientation within our own data in relation to our reference data, which is 1,000 genomes, bringing those 2 data sets together so that we can begin to assess ancestry and select the desired populations that we're interested in using for our analysis. So for our raw data, to give you a sense of what it looks like, this slide is essentially showing you the atypical screen that you get when you import your sample into SVS. And this slide is showing you results from basically a look at our SAGE sample. And basically what you see here are your individuals are represented in rows. Your markers are displayed here at the top, individual RS numbers. You have your marker map info that comes from your map file or your binary map file that you get from Plink. Your sample info, so here this is some sample of SAGE of roughly 284 individuals for 1,040 markers. And of course, within each column, you get the marker calls for each individual. And so, of course, the next step for our process was to obtain the 1,000 genome reference panel data. For us, we wanted to actually filter that on a frequency of about minor of 5%, so we only retain markers, the frequency greater than 5%. Limited the call rate within 1,000 genomes to 99% or more. And since we're interested in using it for determining ancestry at this point, we're going to prune it using an R-squared threshold of 0.5. And of course, once we have that data already in SVS, the next thing we need to do is bring in some annotation information about that 1,000 genome sample. So primarily we're interested in is what superpopulation do these individuals map to? Do they have an African ancestry, Americas, East Asian, European, or South Asian? In addition to that, we bring in the corresponding allele frequencies for each of those populations as well. So once we've done that, we then need to apply our 1,000 genomes marker map containing the ancestry population-specific allele frequencies to our sample data. And we were able to do that using sort of this Python script, right? So we bring the 2 data sets together, filter, do additional QC, limiting the samples to call rates greater than 95% and a minor allele frequency greater than 10%. We then proceed to check strand orientation, and we do that first, of course, by using the ancestry determination sort of frequencies that are sort of based on the average across all superpopulations. So this is what the slide actually looks like when you put your data sets together. Notice here on the left, we have HD representing our 1,000 genomes population. And basically all you're doing, the easiest way to do this is actually to append the two files together because you've already aligned everyone with respect to the markers that you're retaining. So you just append the two files, it'll go very quickly in SVS. It's easier to append than it is to merge, so that would be my recommendation for doing this. And of course, since we're using an LD prune set of SNPs, the number of markers is considerably lower than it was before. So for ancestry determination, you'd actually do that by basically following the tutorial that's on the SVS website. Since you already have your reference population, you follow that tutorial with your data and the reference data merged together. You can proceed to identify, where your samples come from. And so this slide is essentially showing you the relationship of our population, which is shown in orange, to the reference populations. And so here we have the European ancestry population here shown in red, and the orange that's clustered over that is represents our sample. We see some separation as well with respect to this first principal component, which is the y-axis, which is basically largely separating individuals of African ancestry from all other groups, as you can see here. And we see clustering of our data there as well, and we see a smattering of a few individuals in our population to other ancestral groups, most likely. And so if you look at it even further with respect to the second and third principal components, we see some stratification as well, but by and large, a lot of clustering. So for us, really, what we wanted to do was to begin to select out individuals who were largely of European ancestry and largely of African ancestry. And we can go back very quickly to show you, give you a sense how we do that. And what we do really is to define the boundary with respect to the first principal component, where individuals of European ancestry lie and where individuals of African ancestry tend to lie if they're from the reference population. And so if we have a good sense of the spread of individuals along each component, we can begin to stratify our own population using that reference population spread. And so essentially compute thresholds based on the first principal component and use it to separate individuals of European and African ancestry. And then when we want to retain individuals, that falls within 2 standard deviation of the mean of that first principal component. Now, in order to account for any sort of residual stratification that could be going on, we then proceed to use multidimensional scaling using 3 principal components that reside within the remaining set of individuals. And in doing so, we sort of end up removing any outliers with respect to additional components. And what we end up with is a very nice, tight population of individuals of European and African ancestry. So what we end up with at the end of step 1 is much significantly reduced sample sizes for different groups. And what we're showing here really is our ends for the individual samples that were done. So for heroin samples, we have X number of SNPs that were run into that came off those particular arrays, and our final ends. And so we see a decrease there, of course. And then we prepare our samples for going into imputation. So for imputation, we want to subset our original sample file using our determined ancestral groups. So as saw the previous before, everyone came in. We didn't know really where into the reference population. Now we've determined X, Tom, Dick, and Harry now belong to really map onto this particular ancestral group. We're going to carry them forward with our rest of individuals for imputing with respect to European population in 1,000 genome phase 3. So we conduct additional QC and strand orientation checking with respect to that particular population. So we're really redoing the entire process again, but using the reference population of individuals who are only European ancestry. And do the same thing with only individuals who are African ancestry. And then we end up we use SVS to save out each chromosome for those populations as a VCF file, right? So for those of you who may not be familiar with it, we wanted to use a pipeline that was going to be pretty much, say, the new standard within imputation. And so we decided to use the Michigan imputation server. And so much of the protocol for imputation that I talked about so far, it comes from sort of the guidelines that they provide in how to prepare your data to be imputed using this resource. And so this resource is actually supported by NIDA, NCI, and NIH, and it's hosted out of University of Michigan, Gonzalo Becase's group. And so you can prepare your files in pretty much the way I just described here, upload them after creating an account, so you actually get this private environment where you can actually get your genomes imputed. And so we have imputed our data using this particular resource, pulled it back down, selected our 2.2 million markers, and then used those sets of markers for our analyses. And so for our analytical approach, we're primarily using a series of steps, beginning with identifying a common pathway model based on our phenotypic data. And what this involves really is identifying what the factor structure is across alcohol-dependent symptoms in individuals of European and African ancestry. So you can have a multi-group model. Of course, since it's a multi-group model, we need to test for invariance across the different groups. And then we use genomic relatedness restricted maximum likelihood to estimate the additive genetic effect of genotype SNPs within individuals of European and African ancestry, and then between those individuals based on the method that was devised by DeCandia and colleagues back in 2013. And of course, in all these analyses, we include age, gender, study of origin, and account for those residuals stratification using those principal components that went into the multidimensional scaling analysis. And so to basically explain what a common pathway model is, it's essentially a multivariate statistical model that describes the relationship between item endorsements. Specifically, what we're trying to determine is whether a single psychometric factor exists across these items, and in the multi-group case, we're seeing whether or not that factor extends into different subgroups within our data. And so you can imagine then that each of these variables that you see at the bottom, variable 1 through 7, represent the individual alcohol dependence criteria. And for invariance testing, what we really want to know is essentially whether the same construct is being measured in individuals of European and African ancestry. And for that, essentially, we want to conduct what we call the equal forms model, which first tests the number of factors, and the pattern of the factor loadings really are identical across the two groups. Of course, then we proceed to what we call the equal loadings test, which tells us if the loadings themselves are actually equal across the two groups. And of course, within that, we have equal thresholds, really if the item thresholds are equal across groups. And lastly, whether or not we have evidence for the residual variances, meaning variance that's not accounted for by the factor, being equal across the groups. Now, once we've done that, we then take the factor score that we've obtained within each group and use it as the dependent variable in our GREML model. And so GREML really is estimating the SNP-based heritability of the trait, and it's really just this mathematical model asking whether or not the proportion of the phenotypic variance explained by genetic similarity across individuals in the sample is significantly different from 0. And it's implemented in the program called Genome-Wide Complex Trait Analysis, which is developed by Peter Vischer's group. And it's definitely not a gremlin. It's very easy to operate as long as you don't pour water on your computer after the first couple of tries. And basically, you're going to start off by, taking your map files, your genotype files, and estimating genomic relatedness across all individuals in your population. You specify your phenotype file, and the program will do the rest for you. And to give you an example of what the mixed learning model will look like, as I mentioned before, we're decomposing the phenotypic variance into two components: the random effect representing the additive genetic variance, which essentially is for the SNP heritability, which is lowercase g, and of course anything that's not explained by that goes on to become part of the unmeasured environmental variance, or sort of your residual, and essentially your noise. And of course, in our analyses, we incorporate fixed effects for sex, age, and ancestral principal components, as I mentioned before, and those all represent covariates. And in the bivariate case, all we're doing really is extending this model to estimate genetic covariance between, well, not necessarily the two traits, but the same trait across two different populations. And so to summarize the results that we got, we first got evidence to suggest that we have configural invariance across the two groups, but we actually could not constrain the factor loadings to be the same across the two groups. And so pretty much our invariance testing stopped at that point because we had no evidence for equated loadings. As you can see, with respect to the model statistics, both models fit the data well. We have CFI, comparative fit indices, greater than 0.9, and the root mean square error of approximation, or RMSEA, was also less than 0.1, suggesting good fit to the data. And so the circle here in green represents the latent trait that we observed for alcohol dependence in individuals of African ancestry, and the fuchsia or pink circle represents the latent construct that we obtained for individuals of European ancestry. And so we took those constructs further and used them as dependent variables in our GREML model, and we estimated a SNP heritability for alcohol dependence of roughly 31% for individuals of African ancestry and approximately 21% for individuals of European ancestry. From the bivariate model, we actually determined a genetic correlation of 0.76 between the two populations, which is actually quite high. And so, just for clarity, the loading here is representing the standardized loading from a path model, so it's the square root of the overall effect. So you square this, you get the proportion of variance explained. And this is a correlation, so you don't square its actual value. And so what we're seeing really is that genetic variants that contribute to susceptibility to the effects of alcohol are actually likely in linkage to equilibrium with the SNPs that are on these array. And these SNPs are accounting for roughly 20 to 30% of the variation in alcohol dependence. The effects appear to be quite similar between individuals of European and African ancestry. And using the bivariate analysis, we see that there is a genetic overlap between alcohol dependence, genome-wide effects on alcohol dependence, in individuals of European and African ancestry. Now, overall, it seems to be suggesting that both groups have the potential to identify genetic factors that influence risk for alcohol dependence, with each group providing different degrees of power for some individual loci. So really sort of some backing up what we saw in the Learn-Turned-Out study from 2014. Currently, what we're working on really is sort of dissecting whether or not where the source of genetic variance comes from. So we're using a lot of these genome-wide chip arrays and essentially trying to identify the maximal or minimal subset of SNPs that would account for the overall SNP heritability. And just to give some preliminary results from one of our models, using sort of pre-described, effect sizes as mixer distributions, we can actually recover anywhere up to about 10% of the actual total SNP heritability of the trait, with about 5,000 SNPs in individuals of European ancestry and about 1,700 SNPs for individuals of African ancestry. And the difference there is actually quite interesting between the two, given that there's so much more diversity within individuals of African ancestry. What we also have been able to do is actually to recover the entire SNP heritability of the trait using the top 2% of each of our largest mixture distributions. We're still working on that because what we know is that power is limited given the sample size that we're using right now. So as you can see, it's a sample size roughly 8,800 individuals with individuals of European ancestry and roughly 2,000 or so individuals of African ancestry. So we hope to expand the sample sizes here and get more power to resolve where these loci are actually located. And so I'd like to stop there and thank NIAAA, NIDA, and NIMH for contributing to these particular studies, as well as Golden Helix, particularly Bryce Christensen, who was very instrumental in helping us to develop a pipeline that we can implement in SVS to conduct the preparation of our samples for imputation. And thank all of you for listening and take any questions that you might have.
Thank you, Rohan. If you do have questions, feel free to enter them into the questions tab. I will get the recording and slides up on our website and emailed everyone either tomorrow or Friday. More than likely it'll be Friday. We are currently still putting together the details for our next webcast, so as soon as I have everything nailed down, I will email it out to you. And that's about it for announcements. So with that, I'd like to thank Rohan for an excellent webcast and for your time today, and for everyone out there in the audience as well. I wish everyone a great Wednesday.